Good evening. I'm David Levine. I'm co-chairman of Science Writers in New York. I want to thank Joe Bonner, who is behind the scene, who's producing it. My guest tonight is Mirella Salvatore, assistant professor of medicine at Weill Cornell Medicine. She obtained her MD from the Catholic University in Rome, Italy, and completed her internal medicine residency and fellowship in infectious diseases at Mount Sinai in New York City. At Mount Sinai, she also completed postdoctoral fellow training study influenza virus in the lab of Adolfo Garcia Sastre. And she has focused her NIH funded research on the development of novel platforms for influenza vaccines. So Dr. Salvatore, we'd like to turn your camera on.
Hello, everybody.
Hi, thank you for joining us. So if you have a question, please put for the audience, please put that in the Q&A box. The chat box is just for me and Dr. Salvatore and Joe Bonner. So we're going to talk about variants, probably a lot of other things, but why don't you explain what a variant is? And I hear the variants and mutations are used interchangeably. So what is a variant of the COVID-19 virus?
Oh, okay, so let's say that these two terms are related, but not really the same thing. Every time a virus replicates or has to, after infects the cell, needs to replicate. So they will be able to infect more and more cell. And in this replication, we'll need to also replicate this genome, so the nucleic acid of the virus, from which then there are all those blocks that will be translated to form the proteins of the virus itself. And in doing this, in this process, there are sometimes some mistakes. It's like when you type and you make an error, and those typos are mutations. Are mutation that sometimes can bring to a different proteins. Most of the time they don't bring to any variation in the virus itself. Mutation refers to an error in the replication of your nucleic acid, or the nucleic acid of the virus, and this error that is made. Instead, when we talk about variant, specifically in coronavirus, so in coronavirus, is something. Let's say, let me step back. So there is so those errors happen, and for coronavirus, it's thought that they happen. There are 1 to 2 of this error every month that are in the genome. In the case of the variant, those errors will have brought to some characteristics, that some protein changes that have also change a little bit the characteristic of the virus, and they keep to be transmitted. So those, when you have a point mutation, sometimes gets eliminated in time or doesn't bring any effect. This variant that we talk so much, that right at present we mostly talk about 3 major variants: the UK variant, the one that was in South Africa, and the one that is in Brazil, but we don't exclude that more will come in the future, have all a series of those mutations that they keep getting transmitted, and they've changed a little bit the characteristic of the virus. And
so were you surprised that there were variants of the virus, or was that something that you expected?
I work on influenza. So influenza is a virus that actually changed very, very quickly. And I think that those variants are part of this, the evolution. Probably, many people were not. First of all, we didn't know much about coronavirus, but they were not expecting so many variants so fast because the virus has a little bit, has an enzyme that corrects some of those errors. So it's not like influenza that doesn't correct any errors, keep mutating. But you know, let's say, I think everybody was happy that there were no variants, but probably nobody was even so surprised that that happened. And I'm not an evolutionary biologist, but the fight between host and agent and to escape or to improve their viral fitness is something that we see in many viruses and bacteria.
So the big question people have is, are the vaccines that are, so far in the United States, there's 2 vaccines, Pfizer's and Moderna. So then hopefully Johnson & Johnson, AstraZeneca, which is used in the United Kingdom. Are these going to protect us against these variants?
So actually we have data regarding that. So these variants, what those variants, these 3 variants that we'll talk so much, the reason why they became a variant of concern, so as the way I defined, is because there are some of those mutations are in, bring us to changes in the spike protein. And we know the spike protein has an important function for the virus because it's the receptor of the virus, is what the virus, so first is the crown, what give coronavirus the name, is those spikes that are on the surface in the picture that we see, and it binds to the ACE2 receptor of the cell and allow viral entrance. But the other important characteristic is that contains the site, is the target of all our antibody, and is the protein to which antibody elicited by the vaccine bind in order to neutralize the virus, to make the virus not infectious, to prevent the virus from infecting the cells. So that since this mutants have this variant of a concern, have some mutation in the spike protein that is in this place mostly where the receptor binding domain, that is where the RBD, where the spike binds the ACE, and this part the antibody mostly block. So there was a concern that there were that would affect the fitness, the preventive ability of the vaccine. So for the UK variant, so we have several data now, and the first data are from a series of groups in New York as well. They show that in vitro assay, something that we call neutralization assay, using a virus that is not a pseudovirus, those vaccines are effective still in blocking the virus. So they took sera from people that were vaccinated with Moderna and Pfizer, and they tried to block infection of viruses that had the mutation. Actually, they're not the real virus, they're pseudovirus, but they have the spike with the mutation of the UK variant and the South African variant. And they saw that the UK variant is, for sure, there is, in both cases, the vaccine work. The South African variant, they work a little bit less, but still they are good enough to neutralize the virus. And last week we had the first data actually on a population with the AstraZeneca vaccine and the Johnson & Johnson vaccine. Those vaccines were, the phase 3 clinical trial were started a little bit later. And they were both in England and in South Africa. They had some sites in both this region, and the AstraZeneca showed that actually both of them showed that the England England variant is called B117 or 501YV1 variant 1 so there was prevention of the disease. The vaccine were a little bit less effective towards the South African variant, but they were effective in preventing severe disease. There was no severe disease in with any of the in any of the vaccinated individuals. So definitely worth getting vaccinated.
So the bottom line, you should get vaccinated. So if you had to choose between, let's say all 4 vaccines were approved for emergency use, the Pfizer, Moderna, Johnson & Johnson, and AstraZeneca, the Russian vaccine, which one, is there one that you would prefer to take?
The one that is available to me.
Okay.
Like Dr. Fauci said, that the way to prevent those variants is preventing the virus from replicating and creating immunity in the population. All those vaccines are highly effective. Probably the distribution is different based on different factors, because, for example, the Pfizer vaccine, we know that require refrigeration of -80, Moderna -20, while all the adenovirus-based vaccine are more flexible, and they can be at 8 degrees, 4 to 8 degrees. So clearly that make the distribution much easier.
So right now in the United States, it said only 8% of the people in the United States have been vaccinated. Are you optimistic that those numbers are going to skyrocket by the summer?
Well, we always hope for the best.
Okay.
The worst. But I think that today in this conference in the National Academy of Science, there were all the companies, and if we will have the Oxford vaccine, the Janssen vaccine, as the advantage also they require only 1 dose, or at least we have the data at 1 dose, and that is pretty effective, also, we will have the Novavax vaccine as well, the protein-based vaccine. So there are a lot of companies that will bring their vaccines on the market. Plus, there are, I think that Novartis will be producing the Pfizer vaccine. There are other companies that will be helping in producing the vaccine. So I don't know if we get to the 80% that Dr. Fauci is suggesting is the magic number to have this herd immunity, but we always hope that there will be enough vaccine for whoever wants it, and that whoever doesn't want it gets convinced that these vaccines are effective.
So have you been vaccinated?
Yes.
And which one did you have?
The one that was given to me was the Pfizer vaccine.
Okay. Dr. Moore, your colleague, who was on a couple of weeks ago, he's in the Novavax trial.
Yes, I know Dr. Moore very well.
I know. So he doesn't know which vaccine. He doesn't know if he got placebo or vaccine. But
yes, because you see, the difference is that, as you know, that there were the distribution has been by sectors, and so the physicians got vaccinated first, and not all the physicians, actually, all the hospital workers were vaccinated first, and so he also thought that he wanted to get vaccinated before his turn in line. And so he volunteered for the trial. Also, we are running those trials before there was the Moderna.
Okay. So again, I have only 4 questions in the Q&A box. I'm sure you have lots of questions, but also I have some questions that came before, when people registered. So here's someone says there are too many vaccines, too many varieties of COVID. Will there be one vaccine that can be used against all the varieties of COVID? At first, we were led to believe that it takes years to produce a vaccine, but all of a sudden, there are so many of them. Do scientists really work hard to produce them in no time, or politics and profits are the reason for the quick appearance of all these vaccines?
Okay, so too many vaccines, too many variants.
And a lot of several questions right in here. And
well, I can't remember. My neurons
okay.
they have to go one at a time.
Okay.
I will answer those two first. So too many vaccine, too many variants. So first of all, for now, the vaccine are effective against the variant. We don't know what's going to happen, and the company are already preparing for the new variants, but either with the idea of having bivalent vaccine or creating booster with the variant vaccine. The way to prevent the variant from happening is getting some herd immunity, although the viruses might try to escape, like we know the drift of influenza. Yet, I think that companies are thinking about creating vaccines that are universal vaccine, like we've been thinking about influenza, but universal influenza vaccine forever. But I also think that so far we only explored the spike as an antigen that is a surface protein that is therefore highly more subject to variation, and there was no no time they wanted to develop an effective vaccine, so we based what was known for SARS and MERS. But also, none of those vaccines so far include an internal protein and the T-cell immunity. I saw that there are some in development, some live attenuated vaccine that they might stimulate internal protein. They might, and the internal protein are more less variable. So maybe there will be development in that sense to develop broader vaccine that leverage also cell-mediated immunity in addition to antibody responses. Too many vaccine in so short time. There are, first of all, there are two reasons why this happened, or many reasons. So the first is not true that all those vaccines were just created in such a short time. The science behind those vaccines has been developing in years. So first of all, we had before SARS and MERS, and this and SARS came and left, but it was a much more lethal disease that is not COVID. And many companies start to try to develop a vaccine. So we had this knowledge about the spike protein, what you need to neutralize SARS or MERS. And so this was a knowledge that was used now to develop a vaccine for COVID. Then there was this knowledge of the pre-fusion conformation and which mutation are needed to stabilize the spike protein of COVID, because it was derived from other coronavirus. And finally, all those platforms, they are not being created yesterday, but there have been science. The RNA vaccine, there are studies published for rabies and influenza. So the problem with RNA vaccine is that RNA is very unstable. So there have been an evolution. They are known for at least 10 years, but has been an evolution about how to make them more stable. Of course, for COVID, there was a huge investment of money and cooperation, public, private, and a lot of association partnership that allowed these huge trials to be made in short time with cooperation with endpoints that were shared among the trials. So for sure, the investment has been essential to develop those vaccines so short. But I think it's incorrect to say that the vaccine have been fully created in 11 months. We can say that the trials were performed in this time, and we didn't know, but this has built on the knowledge and experience that that company and academia had, and for the other coronavirus.
So here's the question. Will the SARS vaccine reappear each year in various strains as if influenza virus does?
Who knows?
Who knows? Okay. Okay. I heard a scientist says she prefers the AstraZeneca vaccine because it uses live virus, which is an old proven technology. What do you think? Does it use live virus?
No, it's not really a live virus. It's use a live virus, but it doesn't use a live coronavirus. It uses an adenovirus vector. So it uses a virus to carry the information. So when you have a live attenuated vaccine, for example, the flu, the intranasal flu vaccine or typhoid, you have the advantage that you stimulate T-cell responses as well. But I think the mRNA vaccine, they show that they have T-cell responses. So yeah, I don't know. I don't think I mean, to me, the viral vector vaccine doesn't have an advantage in that sense. And if you have you want an old technology, the Novavax vaccine is very similar to what is for influenza. Because it's a protein-based vaccine with the addition of an adjuvant to improve the immune responses. So yeah, I don't know. The study showed that all are safe and effective. So in antibody response. So I personally don't think that the AstraZeneca vaccine is superior to the mRNA vaccine.
So the person says, this is a concrete question. If you have both shots, Moderna, wait 2 weeks and always wear a mask and social distance, can you safely fly from New York City to LA without catching and carrying the virus or possibly infecting the person you're visiting?
Can you repeat the question? So you got.
Here's a person wants to know, if I get both shots and I wait 2 weeks and I always wear a mask and social distance, can I take an airplane ride to LA to visit somebody? Is that safe?
I think it's safe. I mean, because what's the difference to take a plane or to walk in New York subway? Once you are vaccinated, you are protected. What we don't know, but looks like is true, is if this block, if vaccination stop transmission, that is not, if also block transmission. But once one has a mask and has been vaccinated, I think that there is reasonable chances that is safe in taking a plane.
Is it okay? Is there any evidence that any of the variants make the disease more severe or more deadly?
What we know for sure, is that the English variant make the disease more transmissible. In a initial analysis, they suggested that there was an effect on lethality, but they both were equally lethal. But then this English group, actually said that they redid the analysis, and it seems that it's 1.3 times more lethal. But I don't know, the data are not real, are just initial data. It's really difficult with all these epi data also in a system that has been overwhelmed by the disease. So I think that there is no, there is a suggestion that maybe the English variant is more lethal, but we don't know it for sure. Seems like it's more transmissible, but on the lethality we will see when we have published data. For the South African variant, has not been suggested an increased lethality that I'm aware of. And the Brazilian is the least studied of all. I lost my host. How long the vaccine will work in the body? Hi, David, I took your spot.
I'm not really needed. Okay.
Okay. I was just going to the second question on how long the vaccines will work in the body. So how long, that means, I guess, how long are we protected? So the first studies from the Moderna and the Moderna and Pfizer didn't address this point just because they were done. We just needed the vaccine. But there is the AstraZeneca now. I think it's the AstraZeneca. I just heard today they were doing a study to see the persistence of antibody response. Now I give back the work to David. So otherwise,
okay. I actually lost the questions, so you might have to read them. Okay, I lost all the questions. Are there any more questions out there that you see?
Yes.
Okay.
Did you have a reaction to the second dose? I had just a little bit of fever and arm pain, but some people have had some more intense reaction, usually for one day, the second day, and then it went away.
Okay, so I'll ask a question. Joe, can you send me the questions? Thanks. So I have a question. So there's been a lot of media articles saying that you should not take Tylenol or Motrin or Advil or aspirin before you get the vaccine, that it can somehow interfere. And is there any evidence of this?
I'm aware there is not any evidence, but I might have missed one of 6 million preprint that we are submerged with every day. Yeah, I actually read this on some Facebook physician thread that you can take it after but not before. And I kept thinking, I mean, what's the difference? Because it's not that the RNA vaccine is there. We know that the RNA will persist for about a week in the body. So it's not that it goes away.
Okay, so I got my questions back. So if we delay giving people the second dose of Pfizer and Moderna beyond the 3 to 4 week recommended timeframe, we increase the risk that a vaccine-resistant variant will evolve.
I think what we don't know is, one of the things that we don't know is how fast the antibody will go down, and how much antibody you need to protect. Probably for the regular variant, it might be okay, but the concern might be because of, for the South African variant, for which we know that there is a decrease of effectiveness of the vaccine. But the truth is also that AstraZeneca was showing this data that they saw that their effectiveness was increasing if they were given the second dose at 3 months after vaccination. And in UK, I think they approved the vaccine, the AstraZeneca vaccine, as a booster at 3 months. And yeah, so I think we don't know. Maybe I don't even know why they chose 3 weeks or 4 weeks. Usually the booster, yeah, I mean, for many vaccines, we give a booster 1 month. So, but I don't know if they had the time and the possibility to do comparison because everything was so accelerated, of course. And so they chose a time that was a safe time.
There's another news story the last few days saying that people who have COVID don't need the second dose.
There are some, well,
Who knows? Okay.
No, we have some preliminary data actually from Florian Kramer and from Mount Sinai, and maybe also from other people that the first dose will boost your natural immunity, and maybe maybe 1 dose is enough. However, I think, and this is I think that we don't know. The initial response, so what we have learned is that the antibody response to COVID might change according to how severe was your initial disease. And people that had mild disease have antibody response that are not very persistent, while people that have had severe disease have very persistent antibody response. However, also we know that memory T cells, that are the cells that make antibody, will even persist longer than the antibody themselves. I think the real question again is, yes, I give a dose, how much do I need to protect, how much this dose will boost, the second dose, if I had COVID, will boost my antibody response, and for how long? Do we have this data? And I don't think we still have the data for how long this will persist, considering that there is a variability.
Well, the concern was that some people who were getting the second vaccine, who had COVID, got very sick from the second vaccine. Their symptoms came back. So who knows? Well,
the symptoms came back. Well, they got sick because probably they had an extra response. But I think that they will be protective from COVID. While in the opposite way, you might not protect, and you might increase the variance, because the problem is that if you are not, you don't have an antibody response. So let me digress 1 second. So one of the hypotheses that was done, how this variant started in England, is that the variant come from an immunocompromised host. And we know that in the immunocompromised host, the virus can replicate with suboptimal level of immunity. And so the virus has the power to circumvent the immunity that the virus has. And maybe, so if we vaccinate and we don't have a good response, maybe, maybe we will increase the number of variants that will try to circumvent the vaccine response.
So are you worried about newer, worse viruses coming in the following years? And are we doomed to always wearing masks? Okay.
No, I don't think so. I don't want to think that we are doomed to always wear a mask. Probably we'll get to some, I mean, if it's true that the vaccine will get some level of protection. I don't know. My hope is that we will get to something like other viruses, correct, where we get some level of immunity. And yes, there will be always some, even with flu, we know that there are some people that do worse than others, but there is a good amount of population that will get enough immunity to be protected. But of course, I don't know. What was the other question? If I'm worried about the variant, we are always worried, not only about the variant, but I think that what we have to There is this concept of One Health, and One Health is the relationship between environment, animal viruses, and human health. I think this is very important and sometimes unappreciated. We don't think enough. And all this coronavirus came from bat through an intermediate host. We know that bird flu comes from bird. We know that yellow fever came, the outbreak that was in Brazil came from the deforestation of the Amazon and the monkey that moved from the Amazon to the city. So I think that I worry, but I worry in a more general complex, not necessarily about the coronavirus variant, but there is so much that if one think about, there is so much to worry. But worry is not a positive, it's not a positive sentiment. I don't know, I don't have the English word right now, but I think that more than worrying, we need to be proactive and have a solid science to study these issues.
So how much protection does the first dose of Pfizer vaccine and Moderna give you?
I don't know if they have the exact, they calculated the protection after one dose. I don't know this answer.
Okay. So can you talk all about the recent AstraZeneca Oxford data that seem to indicate reduced transmission in vaccinated individuals? Is it fair to say this is truly reduction in transmission versus just reduction in infection?
I haven't seen the original data, so sorry, I cannot comment. I was saying that today there was this very interesting meeting, but unfortunately I couldn't attend fully, and I missed this part.
Okay, so if you have more questions, please put them in the Q&A box. Earlier, you mentioned the possibility that vaccines could target internal viral proteins that would be less subject to mutation than the spike protein. How far along are those?
The only, so I know that there are a few vaccines in stage that are going in phase 1 trial. I saw there is a live attenuated vaccine that is going in trial. It doesn't mean that they only target the internal protein, they also target internal proteins. I think that I wanted to clarify that, because cell-mediated immunity by itself often is not enough to prevent infection. Help clearing the infection, but not preventing it. So usually, and maybe I apply flu concept to COVID now, but for example, for influenza, if you have a vaccine that target only the nucleoprotein, you can, they work beautifully in mice, but they don't work in human, because you need antibody against the hemagglutinin.
So you've had 2 doses of vaccine. So have you changed your habits?
No, I feel just relieved when people don't wear masks in the subway.
Okay. So have you been taking the subway throughout the pandemic, or?
Yes, and I was negative before the vaccine, so something I did right.
Okay, so subways are not really a major risk.
I think there was nobody in the subway when I was taking the subway.
Okay. Okay.
I also was actually, during the pandemic, I was so paranoid. I was wearing an N95 in the subway, and leaving my clothes outside of the house and jumping under the shower. But yeah, spending most of the day in a hospital also, probably the subway was much safer.
Okay. When one talks about 80% vaccinated for herd immunity, does that include children? And what is the age range that is assumed?
Okay, so actually this number just the other day, Fauci explained how he got to this number. This number estimate actually came from the measles vaccine, and he said that measles is very highly transmissible, much more transmissible than COVID, and the vaccine is also better than COVID. And for measles, you need about 70 to 80%, and that's the way he got to this number. Now we know that children have a different distribution of the ACE receptor, and they get less infected. I don't know if we have the good data on the transmission of the children, but we don't have a vaccine for the children. So I think that for now we stick to the 80% of the adults.
What should children be vaccinated?
Should children be vaccinated?
I mean, they get flu vaccines.
Yeah, but they die for flu. Why they don't die for COVID? They should get vaccinated if it's proven that they are, when there will be a vaccine that is approved for children, because there are no studies in children. The same that there are no studies in immunocompromised or studies in pregnant women. I think the youngest age is the adolescent, but not children. If there is a vaccine that is safe for children, I think it would help in the transmission to the elderly, because maybe or immunocompromised individual that might have an immune system that might not fully respond to the vaccine.
Do you think our society can get back to semblance of normal without children getting vaccinated? Could we end the pandemic without a vaccine for children?
Who knows? Probably, possibly. You know, there are so many factors. I think that before thinking, there are so many problems right now in distribution of vaccine. We will not be able to get rid of the virus until I think we vaccinate underdeveloped country too. And so I don't know what's the status of the vaccine distribution in some of those countries, and what are the plans. I know that there is a lot of talks about equitable distribution, but clearly there are challenges about sites where people can get vaccinated, about carrying the vaccine there, about cost. But so those clearly with travel and if the part of the world doesn't get vaccinated, that's probably more worrisome than if we are here and everybody gets the vaccine. I think that for us, a lot of challenges are from vaccine refusal.
Okay, basic question. If I get the vaccine, how long is it going to give me protection for?
Well, yeah, that's what I say. We don't have data on this yet, because the study were both Pfizer and Moderna will follow the individual for I think 2 years. They said there are some data from AstraZeneca that is collecting. We don't know how long the protection is going to last, and also will depend if there will be some different virus that the vaccine doesn't succeed in protecting. But for now, I think that they have data 8 months.
So we have to get,
I mean, 8 months doesn't mean that it cannot be longer. I mean that we got up there.
It's a new vaccine. So do you foresee that in the future we'll be getting yearly, COVID-19 vaccines as we did flu vaccines?
Who knows? Maybe 1 year, maybe 3. Maybe we can get a booster every 5 years, like we do for tetanus. I just don't know. I think we don't know yet.
So you said that some of the companies are working on different vaccines to cover the other variants. So do you foresee that in the future there will be better, just like shingles. The first shingles vaccine is now replaced by the Shingrix vaccine, which is 90% effective. And the first one was 50%. Do you foresee that there's going to be better COVID vaccines?
That's called evolution of science. The effectiveness of those vaccines is very high. I mean, as a matter of fact, we have to see also if now on population, what are the real number of the effectiveness when we vaccinate not the people in the clinical trial, but the regular population. The fact that the clinical trial made a special effort to include people with comorbidities and several ages, I think that is very positive, but I think that, you know, the persistence for now is the question, the persistence and the cross protection with variants, with future variants.
Okay. How long does it take the body to mount a good immune response after vaccination?
Usually it takes, depends if is the booster or not the booster. The first dose, it takes about 2 weeks to 2 to 3 weeks.
And the second dose?
Well, you might have a faster response because you already have the cells that are they have seen the vaccine, so you might get 10 days, 7 to 10 days.
7 to 10 days. Okay. Should we be doing contact tracing on people who get vaccinated in order to determine whether they can spread the disease?
Well, the company are doing some studies regarding the ability of protecting from transmission.
So there's a story on the news how in Britain they're doing genome testing to see what's going on, and that we don't do that here at all.
No, we are doing it right now.
Where are we doing it?
My collaborator.
I know Stanford because that was in the news today.
Well, my collaborator NIH. I think that I read a program is called SPORE, but I might making this up. That is that are collecting that have some center doing genomic sequencing. There is also today. I mean, we haven't done until now, but we are starting doing it. And again, we know how to do it because we do it for flu and for new vaccines. So it's not that there are not the capability to do it. And yeah, so, and maybe in the future there will be also some, there are under development some tests in which you will not have to sequence all the genome to look for some specific mutation, but surveillance with sequencing of the whole genome is necessary to make sure that we know the variant that are circulating and if this will recombine, because this virus might recombine and create new variants that are of concern and can affect more than the vaccine, maybe the monoclonal antibody responses.
So is there any thoughts to communicate with someone who is a vaccine denier or hesitant about getting the vaccine?
There is any possibility to communicate, apart for doing all the education and keeping talking, and I think that there is a lot of attempt to outreach and educate. I think that the concept that scared a lot of people is the how fast those were developed. And, you know, we keep saying how fast those vaccines were developed. And I think it's not clear that to many that, yes, it's fast, but it's fast because there were the money and there was the science that were already developed. So it's fast because we could recruit 40,000 people in, in 3 months by using all the HIV, for example, side network to enroll patients, and there was a real very strong partnership, public and private, and BARDA and NIH and the all institutional sites.
So I like to end by asking people what they do. And I guess with you, it's pre-vaccine and post-vaccine. So did you go to restaurants before you had the vaccine?
Me?
Yes, or do indoor dining?
I cook.
You
I'm
cook.
Italian.
Oh, Italian, okay.
No, I go to the restaurants. I always thought that if the restaurants, not so much, just because, yeah. But I'm really missing my gym. So after vaccination, what I was thinking, like, oh, maybe I can go back to my gym.
Okay, so is it safe to go to a gym after you've been vaccinated?
Well, gyms are open.
I know they're open, but you didn't go, right?
I didn't go because they are not having classes. But no, I also, we are way too busy, honestly. Yeah, I think that after we get vaccinated, at a certain point, although we are concerned, but when some level of vaccination of immunity are around, we will have to, start again some sort of normal life.
Okay.
And again, there was no case of severe disease in vaccinated individual that has been reported.
So have you flown anywhere since this started?
Me?
Yes.
Yeah, I flew during the summer. I went home. I went to Italy.
Okay. Did you wear an N95 mask?
I did, but not my husband. I couldn't convince him.
Okay. But he's okay.
Well, also the airline, there are many airlines that are doing this COVID tested planes, in which you get tested the day before embarking the train, and then get tested again when you arrive. I think that well, the variants are already here, but I think for international travel, maybe that's a good way to prevent spread through countries, although I don't know how easy it is.
Okay, so this weekend is the Super Bowl, and the CDC is warning not to have parties, not to scream and yell, blow a like a New Year's Eve thing, clap. I mean, is this practical advice that we're getting from the CDC, or is this kind of,
Well, we have seen after any holiday, we saw an increase of cases of COVID admitted to the hospitals. So it's true that this happens. And I also, in Italy, we had the second wave that was very bad. And what they decided to do, they had a very strict what do you call? They closed everything on Christmas, so people couldn't even travel from a region to a region. Much more severe than here, and the number went down. So we know that those public health measures really work. If they're practical, I think we are all fatigued by having COVID for 1 year, and has changed so much our lives. So.
My friends in France say that they have a curfew after 6 p.m.
Yeah, they are. I mean, they really the restaurant and the bar closure and everything. You know, in Christmas, everybody wants to go in the house, and you couldn't go unless were the people that were your strict relative and you were living with them. And even for walking in the street, they had like all a series of restrictions. Can cross the region border. All the Christmas markets, they were canceled and masses. And so it was a pretty big deal. But the numbers went down, so are going down.
And the numbers are going down now after the post-holiday surges. So it's good.
Yeah, actually, it was before the holiday, and then they closed everything, and then the number are going down. Yeah. Okay. Also in New York, I think we are in a plateau going down.
Okay. So next week, my guest is going to be Apurva Mandavili. She's a reporter for the New York Times. You see her every day. She has a headline. She's one of the chief COVID reporters for the New York Times. So, Dr. Salvatore, I'm sorry that I lost my Wi-Fi connection, but I want to thank you for spending an hour with me. It was delightful talking to you, and it was nice talking to you before the show as well.
Thank you so much.
So thank you very much, and everyone stay safe and try and get a vaccine appointment. I know it's not easy.
Definitely.
And stay,
hang out a few more months, hopefully.
And stay safe and wear a mask. Okay, take care and good night.
Good night.
Okay.